Okay, here we are. Vanessa Gordon wearing a hoodie. 22 people. How many people are in this class? About 30. Stefan McCain, my man, he's here. Elijah Crossborn. Crossborn? Mm-hmm. Mm-hmm. What do we got now? We're up to 24. Let me take a quick look to see how many people are in this class, just to see how close we are. Mm-hmm. Mm-hmm. Restro. Restro on the rester. 35. 35 people in this class. Okay. Okay. We've got 26 here. Hopefully they'll catch up on video later. All right, so why don't we just go ahead and get going. I will start by sharing my PowerPoint. PowerPoint. Where's my PowerPoint? There it is. Okay. Slide. Boom, 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 boom. We're doing chick today. Chick development. So we'll see, this is quite a bit different from the frog, maybe a little bit similar to the fish, but the frog had a big hollow ball of cells, the blastula. Here we're going to talk about a blastoderm, just a thin layer of cells, like a layer of skin. That's why they call it a derm. So we'll start out with the blastoderm or blastodisc, same thing. Cleavage. We end up with this kind of disc thing with a clear area in the middle and an opaque area around the outside. And that will then gastrulate via the primitive streak and then nerulate back down that primitive streak. And we'll end up stopping after nerulation. I think that's about as far as we tend to go with these models in this class. So anyway, fertilization in the chick, as you probably know, is internal, right? So, you know, again, that was a nice thing about the frogs and the fish. We could do it experimentally in the dish, and it was pretty easy to do. Here, this is internal to the chicken, and you end up with all the cells on top of the yolk. Here's the yolk. These things here are just kind of stabilizing the yolk so it doesn't keep rolling on you, or on the poor old chicken there. So we've got the blastoderm on top, the yolk egg, the yolk here, which is where all the energy is going to come from. Here's the egg white, which is where all the protein is going to come from to build that chick. And the blastoderm is on top. I guess that's all there is to say here. Okay, so here's what's happening in the chicken after fertilization. You start to get the cleavage furrow. So this was the original fertilized egg cell. And you start getting cleavage furrows, but the cleavage furrows don't go all the way down through and form individual cells. So at this stage, you still have a syncytium. Again, one of my favorite words of all time, syncytium. So you've got a multinucleated mass of cytoplasm here, that is syncytium. Shortly, those cell membranes will complete and each nucleus will be in its own cell. But right now it's a syncytium. Anyone ever hear the word syzygy? Another one of my favorite words, syzygy. S-Y-Z-Y-G-Y, something like that. It's a perfect alignment of 3 stars. Just a good word. Comes up in spelling. Okay, so the next stage then, the cells start to complete themselves. So you've got individual cells, and you get the formation of this subgerminal space. So germ is always like the cells that are growing. So this is underneath those cells. So subgerminal space. And this is really important. This will start to get us into the embryo proper here. So now this is, no, let's wait till the next stage. Okay, so at this stage, you've got this space here and a double layer of cells on top. As this goes to the next stage, cells keep dividing and dividing, and you end up with cells being thickened at either side. And you've got a thin layer on top, and in the middle, there's really nothing but yolk. So if you look at this from above, the center is translucent. So that's called the area pellucida. Okay, this whole area where you've got space. And then on the outside where you've got thickened areas, that's zona opaca, or opaque zone. And then these cells will continue to divide. And in the next stage, you've got cells all across the yolk here. You've got your thickened edges and cells across the top. So now we've got a real differentiation between tissues at this point. These cells at the bottom are called hypoblast. Hypo means under, right? Under the blast, or the growing embryo. And this is epiblast, or surface blast, right? So this is under and this is the surface. This hypoblast now will not become part of the chick in the end. This is what we call extra embryonic tissue. Okay, that's a good word to know too, extra embryonic tissue. So this tissue will, it will develop into structures that will take, the umbilicus basically, structures that will take nutrients from the yolk and oxygen from the outside of the shell. So that's going to develop into those extra embryonic structures that support the embryo's development. And then the surface cells, the epiblast cells, are what are going to actually become the embryo itself. Okay, so at this stage, when you've got this subgerminal space, your hypoblast, your epiblast, this is when the egg actually drops out of the chicken. So this is when laying happens. So here, right about at laying period, this is your zona pellucida, zona opaca. Right here at this edge, you get a thickening of cells that's called Kohler's sickle. Okay, so you've got a dense bunch of cells here. And we'll talk later about what those cells are doing, but basically they're releasing hormones to induce cells to change cell type and to activate, gastrulation and stuff. So what's important to know now is where this Kohler's sickle forms will be the posterior end. So just to kind of keep reviewing as we're going through, when we were looking at the xenopus, at the egg stage, we only had one axis, the up and down, right? And we made that anterior posterior axis by gastrulating. Here, gastrulation hasn't happened, but because it's a flat disc, and because of the way gastrulation happens here, we pretty much already have all 3 axes ready to go. Okay, so you've got your posterior end will be here, your anterior end up here. You're looking down at the dorsal end. If you look up from the bottom, you'd be looking at the ventral, and then you've got right and left here. Okay, right, left, dorsal on top, ventral underneath, anterior, posterior. And that's the way it's going to develop from here on now that the egg has been laid. Okay, so gastrulation is going to start now. Gastrulation starts at Kohler's sickle. and you get this primitive streak that's going to go up from anterior to posterior. Okay, so you've got primitive streak formation moving forward, or in other words, towards the anterior end from Kohler's sickle. Now, what this is exactly, this primitive streak, is the fact that these epiblast cells are dividing. And when they divide, they push forward into the center and they crawl through this little streak and then into that subgerminal space. And they're going to stay associated with this epiblast cells, but they're coming down in and underneath them. Now, what's really important about this is a number of things are really important about this. One is once these cells go through the primitive streak, they become mesenchyme cells. So mesenchyme is an important word. We'll see this quite a bit throughout development. Mesenchyme means they are loosely organized, so they're not attached to each other like a tissue would be, kind of individualistic cells. So they're loosely organized and they're motile, so they're going to be moving, crawling along things. So I think I said this the other day with the neural crest cells. They're mesenchymal cells too. Okay, so these cells that come through, number one, they're mesenchymal cells and they will crawl. Oops. Number two, they were epiblast cells that divided, and when they came through the primitive streak, they got induced to become both mesoderm and endoderm cells. So some of these end up mesoderm, some of them end up endoderm. Okay, so this is where we get specification of mesoderm and endoderm. Okay, so this streak now is moving forward in this direction. Let me, hang on a second. Let me stop this share for a second. I want to take my background off. Can you see me? No, not really. I'm kind of all washed out. I didn't get a better background. See, the problem with the virtual background, I guess that's good enough. Problem with the virtual background, you can't see my hands. And while I'm not Italian, I still like to talk with my hands. Okay, so the reason I'm trying to get you to see my hands here is this primitive streak is moving towards the anterior side. Now, as it's moving forward, cells are coming in through the streak and they're moving forward and sideways. Okay, so imagine these cells, they are moving towards the middle because the cells on the surface are dividing. As you get more cells, they have to go somewhere, and they go towards the middle. They come down in, and as they go through that streak, they become mesenchymal, so they separate from each other, and they go forward and laterally. Okay, so you're going to fill out this whole subgerminal space with the endoderm and mesoderm by these cells coming through this way. Does that make sense? Stefan, can you give me a thumbs up, thumbs down? Thumbs up, okay. Okay, so that's gastrulation. Oops. And gastrulation, again, starts at the posterior, goes all the way to the anterior end. And you can see the embryo is kind of elongating. It used to be a disc, and now it's more of an oval shape. Oh, and again, and at the same time, these are specifying into mesoderm and endoderm. These epiblast cells are becoming ectoderm. So now you've got all three types of tissue there: ectoderm, mesoderm, and endoderm. Okay, when this primitive streak makes it all the way to the anterior end, it becomes Henson's node. Okay, so you've got a node here. And this node is basically, again, a whole lot of cells dividing and going in. And it's obvious, there's so much activity going on there that you can see it in a light microscope, and it looks like a node, so boom, there, node. And if you probably could imagine who discovered this node. Henson named it after himself, egomaniac. So you've got this Henson's node here where cells are coming in. So we're about to start neurulation. Right before neurulation starts, the cells that are going in here are still going anterior and lateral. So you've got cells that are coming in through this node and moving forward and moving sideways. Okay, so they're filling in that direction. And these cells that are going in here at this early stage are going to be head process and precordial plate mesoderm. Head process and precordial plate mesoderm. So the head process will become skull, and the notochord comes out of the head process. Precordial plate mesoderm are other, facial mesodermal structures. I should know that. Wait, let me give you a little more detail on that if I can find it quickly. All right. Oh, shoot. Ooh, vocabulary word I didn't say, I'll have to remind you. Okay, so after the primitive streak has elongated to its full length, some cells from Henson's node start to migrate forwards along the midline and under the epiblast to give rise to precordial plate mesoderm and head process. The head process is the emerging rod-like notochord, and the precordial plate mesoderm is looser mass of cells anterior to it. After the head process forms, the regression. Okay, so precordial plate mesoderm is anterior to the notochord, and then the head process becomes the notochord. So notochord, I'm hoping notochord means a lot to you, but at this point it may not. Basically, notochord is going to be mesoderm underneath the ectoderm that will turn the ectoderm into the neural tube. So notochord is really important because it's going to organize that neural tube. Okay, Henson's node. You still have cells going in, going forward. They're going to be forming the head. And then this Henson's node now is going to move posteriorly. So I like to say it's like gastrulation zips it up and neurulation zips it back down because the primitive streak goes up and then Henson node comes right back down. So as it starts to come down now, it's going to be laying down notochord and somites. So Henson's node is like the Spearman organizer in the xenopus. Okay, it's going to be an organizer, which means it's going to release hormones that will specify tissues to be different types of tissues. Stefan, is that whiskey you're drinking in the middle of the day? No? Okay, good. I just wanted to make sure. Okay, so primitive streaks then starts to regress posteriorly. The Henson's node leads the regression, and it's going to be laying down notochord and somites. And the cells for this are all coming from this front end of Henson's node. So this is called the stem zone. Stem zone. So basically like stem cell. A lot of cell division here, producing new cells, and these cells are going in through Henson's node and always going anterior and lateral. And they're producing, the ones that stay in the midline are becoming notochord, and the ones that go kind of lateral more will be somites. I've got a chat question. Don't lie, Steph. Stefan McCain. Okay. Okay. So that's going to continue to go downwards, goes towards the posterior end. And here you can see the head is starting to form. Starts out called a head fold. Here in this figure, you can see the stem zone. And again, this is where cells are dividing, and they're going in through Henson's node and underneath. So they always have to go under because the surface is going to be ectoderm. So these cells are coming in, becoming mesoderm, and underneath notochord and somites. And once that Hensen's node gets all the way to the end, it stops, and everything that goes below that, all the cells below that become the postanal tail. Now here you can see the somites have formed, and the somites will be and then neurulation will produce the spinal cord, and somites will be the the bone around the spinal cord, the vertebrae, and then the lateral parts of the skeleton of the body plan. Okay, so here's what that Hensen's node with stem zone is like. I made this picture myself. I thought it made things easier to see, but I'm not sure. Okay, so here's your stem zone here. So you've got cells dividing out here. They're coming over around and then diving down in. And the ones that are in the center here become notochord, and the ones that are more on the outside become somites. So you end up with notochord in the middle, somoid at either side, and then the ectoderm across the top, and that will now neurulate. Okay, so neurulation then occurs just like it does in the other models that we looked at, in that you get these neural folds coming up. You've got a neural plate right on top of the notochord, and the neural folds come up and they bend over, and eventually they meet at the top and close. And once they close, you get the neural crest cells that will then move out of there and go on their merry way. So in the end, you end up with your neural tube, epidermis across the top of everything. Okay, that was ectoderm, now it's epidermis. You've got your neural tube, your notochord here, somite to the left, somite to the right. And then we saw a schematic form of this in the last class, I believe. So now you've got, this is all mesoderm over here. This is lateral mesoderm. And the lateral mesoderm is divided into somatopleural, right? And the somatopleural will be like ribs and muscles and limbs. And then you've got the splanchnopleural, hate that word, I don't know how to pronounce it, splanchnopleural, which will be inside, and that will be like heart and other internal mesodermal things. Not gut, remember gut is endoderm, but everything else that's mesoderm on the inside will come out of this splanchnopleural thing. Okay, so now you've basically got a flat sheet of cells here with all three different types of primitive tissue, and now you've got even more specification, because this isn't just ectoderm anymore. Now it's neuroectoderm and epidermal ectoderm, and your mesoderm is starting to be specified as well. You've got somites, intermediate mesoderm, and lateral mesoderm, and two different types of lateral mesoderm. So we had a lot of specification happen at this time. You also have endoderm across the bottom of all of this. Okay, so this lower layer here is all endoderm. Now what's going to happen is this flat looks like a planarian. It's just flat. It's going to curl up. So the inside parts of this are going to curl together and close. So this endoderm curls together and closes, and that's going to form the gut. So under the head fold, you have an endodermal pouch that will fold up, and that becomes the pharynx and the gut, the foregut. In the posterior end, same thing, folding up, and that's the hindgut. And in the middle, the sides of the embryo will fold together and form the rest of the gut. And the whole thing will close up. The whole gut tube will close up from either end, except for the very middle. In the very middle, the gut tube doesn't close up, because that's where the umbilicus is going to come out and interact with the non-embryonic parts of the egg. Okay, where are we?
Is there a difference between like when eggs like like when you go to like Walmart to buy eggs to like cook and these type of eggs?
That's a good question. I've always wondered that. What I've heard, and I'm not going to put any money on this, is that the ones we get in the store are unfertilized eggs. So I guess once the chicken starts laying eggs, they just keep laying eggs whether they have sperm or not, and they're unfertilized. That's what I think is true. It's possible they're just young eggs that haven't developed very far, but after a couple of days, you start to get structures that you would see in the embryo. So it's probably more likely they're unfertilized.
Okay, thanks.
Sure. Um, Size of the embryo fold together. Okay, so the size of the embryo formed together form the gut, and the rest of that mesoderm is going to form around it to make the body cavity, and then limb buds are going to come out, and you're going to make limbs. Also, from the mesoderm, you've got a heart on either side. They're called heart primordia, and they will move to the center and fuse together to make a four-chambered heart. Um, and they fuse together at the midline under the gut, just like where your heart is. Um, and then blood vessels and blood islands will form in the umbilicus, which is extra embryonic tissue. But then they'll fuse with the embryo, and that becomes the circulation for the chick. Okay, questions? The thing about this stuff is you got to look at it a bunch of times before it starts to make sense, because it's all just words on a paper, and you have to find a way to convert that into images in your mind and try and imagine what's going on here in 3 dimensions. Let me just go over it real quick one more time. So you start out with a whole bunch of cell membranes forming without completing. So this is all just cytoplasm with multinuclei in them and some membranes that haven't sealed up. And then those membranes seal up, so you've got individual cells. And once that happens, you get this space. It's going to be important for gastrulation. That space opens up further, and cells start to grow across the bottom, across the top of the yolk. You've got a clearish area in the middle, which is pellucida, and thickening at the sides, which is opaca. Eventually, those cells totally cover the yolk, and that's the hypoblast, which will be extra embryonic. And then you've got epiblast, with the cells on top, which is where the actual embryo will come from. Everything comes from this, because at the next stage, you start to see these cells divide, and they sneak in through, and they form the 3 layers of the embryo from there. And this is all happening as this primitive streak is going forward to the anterior side. Once it hits that anterior side, cells keep going through that node. They form the head process, and then the node regresses. And as it regresses, it lays down the notochord, and that causes neurulation to happen, because the notochord induces the ectoderm to become neuroectoderm. The Hensen's node reaches the end, and it stops, and then you've got your postanal tail. Okay, someone's chatting me up here. What causes the subgerminal space in the first place? What makes that open up like that? I don't know. I could guess there's probably some kind of molecular mechanisms pumping water in there. So let's see here. It's all part of the same cell with no cell membranes, but once you get a membrane formed on these cells, maybe they're pumping water from the outside into the space. I don't really know. I imagine it's a fluid-filled space. Yeah, that's a good question. What makes that happen? Okay, and then neural folds, neural tube closing up. 1, 2, 3, and then it's totally closed. Okay. So other than that, we've got our extra embryonic structures that are also important, very important in both avian and mammalian development. We've got the amniotic cavity, which is where the embryo is contained, right? Amniotic fluid in there. Around the outside, the membrane that holds that together is the amnion. Okay, so the amnion holds that amniotic fluid together. Now, part of the umbilicus kind of goes two different ways. On the one side, it comes over here to the allantois. So the allantois are kind of like lungs. I mean, they're not lungs. They're not where the lungs are coming from, but they're external tissue that acts like lungs because you get blood coming in here, and then this allantois comes really close to the chorion. Chorion is a membrane that goes around the whole thing inside the shell. And then you've got some air and carbon dioxide exchange here. And then you've got the umbilical artery bringing oxygenated blood back into the embryo. So that allantois then is kind of acting like lungs, although it's not developmentally going to be lungs. Here's an interesting thing. So this is really important to be able to get oxygen and lose CO2, right? So Lake Champlain has got this invasive species, the cormorant. Maybe you've seen them. They sit really low in the water, so it looks like a snake with its head sticking up. It's kind of a funny bird. And when it flies, it tends to stay really close to the water as it flies out. It's all black, kind of looks like a duck, but the neck is a little bit longer. So these are invasive. They're not supposed to be here. They never were here anyway. So they're causing a lot of ecological damage. So one year, they decided they were going to get rid of them. And there were so many of them that would nest on one particular island that their poop killed all the vegetation on that island. So it was like an island that was totally denuded except for nests and poop. And so the biologists went up there and they broke all the eggs. Okay, that'll get rid of a whole generation. Well, no, because the cormorants came back and they laid more eggs. You know, once their eggs were gone, they laid more eggs. So what the scientists decided to do was they took oil and they went there and they oiled the eggs, because when they put a layer of oil on top of it, the egg couldn't exchange gases anymore. And so it died. But the cormorants didn't know they were dead because they weren't broken. So they sat on dead eggs all summer and they lost a generation of chicks. I'm not sure how well that project went in the long run. This is probably, I don't know, 5, 6, 7 years ago. But that was kind of a neat trick they did to trick the cormorants into not having any more babies. But anyway, point being, allantois, gas exchange. The other important part of the umbilicus here is the vitelline artery and the vitelline vein. So the vitelline artery is going to bring blood down into the yolk sac and pick up nutrients. And then the vitelline veins will bring nutriated, oh, that's a good word. I think I just made that word up. Nutriated blood back into the embryo. Okay, so here, extraembryonic structures, we've got the amnion, amniotic cavity, vitelline artery, vitelline vein, umbilical artery, umbilical vein, the chorion, and the allantois. And remember, arteries always have oxygenated blood. It doesn't matter what's going towards or away from the embryo, it's always oxygenated blood. Okay, and that, my friends, is all we have for the chick. Time is it? So we've got 15 minutes left. I think Monday we're going to have a quiz on what we talked about this week. It's not going to be graded unless you do poorly, then I'll grade it and subtract points from you. But we'll give you a quiz on Monday because I feel like every class I'm going to have extra time. Maybe we'll quiz you more often here and make sure you're up on things. So that's about it for what I have today. Any questions?
Can you repeat the heart stuff you said before?
Which heart stuff?
You talked about the heart formation with the mesoderm, and I didn't catch all of it.
Okay, so these notes are posted up there in Moodle, by the way, if you missed something. But what happens there? This is at the point where neurulation has occurred, and now you've got lateral mesoderm, and it starts to surround the gut. And as it's surrounding the gut, it forms two hearts, two primordial hearts, a left and a right. and they move to the middle and they fuse together to make one 4-chambered heart. So they're called heart primordia. Heart primordia, one from either side. Okay.
I have a question. So you said extra embryonic tissues used to form the shell?
No, the extra embryonic tissues are the vitelline artery and vein, the amniotic sac, the amniotic membrane. Hang on, let me get that back up. So it's not necessarily the shell. I guess the shell is extra embryonic too. It's basically everything except the embryo itself. So the amnion, the amniotic cavity, the chorion, the allantois, and the yolk sac, that's all extra embryonic. Okay?
Okay, thank you.
Okay, can I see all your faces real quick before I say goodbye? So I don't feel so alone. Sammy Robinson's here. Good to see you, sir.
Good seeing you too, Dr. Slish.
Antonio Estevez. I don't think I've seen you before. Good looking kid, Anthony. You know, if you were from Philadelphia, you'd be Annie.
Why is that?
Be Annie. Stefan McCain. Yusef Okine, there he is. Victoria. Michelle Simmons is shy. That's okay. Kwame, my friend. Yeah, I've had you every semester, I think. Every semester since you've come here. It's good.
Yep.
Kwame's another one of those students who's going to get a master or a minor in Slish. I think Maria has got a minor in Slish coming. Okay. Evans Cujo's working on his minor in Slish too. I had you in 4 classes already. There he is.
That's true.
Nice beard, son. I was going to grow a beard and my whole family told me not to.
Really,
So,
it's good. Well,
okay.
Thank you for the thing. I got an explanation.
No problem.
Yeah.
Okay, so I'm going to let y'all go. Hopefully we've had fun today and we've learned a little something too. So Monday, we'll make sure you study up because Monday there'll be a quiz. Maybe I'll make it worth bonus points so it's something anyway. All right, see you then.